Most years I've come up here and presented this, and I just jump right into the Farquhar model. And I know that Pat gave well, I don't know that he did, but based on everything I know from Pat, I'm absolutely certain he gave a very good overview of photosynthesis yesterday. But I just didn't want to assume, going into this, that everyone understands the very basics of photosynthesis. Since you're taking this course, there's no doubt that you understand the importance of photosynthesis. This is just some iteration of the global carbon cycle. These numbers change quite a bit from year to year, from researcher to researcher. But ultimately, if you look at this carbon cycle, this 121 gigatons of carbon entering the terrestrial biosphere, is a massive component of the global carbon cycle, and it represents somewhere about a fourth or a fifth of the total carbon dioxide pool that's in the atmosphere. And before I get involved in the chloroplastic modeling of photosynthesis, I thought I would present what I unashamedly stole from Dennis Baldocchi's, one of Dennis Baldocchi's lectures online, to basically talk about how we arrive, one of the ways we arrive at this 120 gigatons of carbon entering the terrestrial biosphere, or the gross primary productivity. And this is based on, well, basically a lot of assumptions as well as some constants. So if you have the solar constant, which is about 1,300, a little over 1,300, thanks. A little over 1,300 watts per square meter. And if you average this over the Earth's surface, because the Earth is a sphere, it works out to be about 341 watts per square meter of solar radiation at the top of the atmosphere. And then you assume that you're going to get some losses from transmission through the atmosphere. And of course, all this radiation, not all this radiation is usable. So only about half of it's usable via photosynthesis. And then you have to convert the energy from watts per square meters to photons, and this is just a conversion factor. And photosynthetic efficiency, this is, about a little less than 2%. Right? So on a terrestrial ecosystem, at best, we could get up to 4% efficiency. But the reality is most natural ecosystems see significantly less than that. So we're assuming it's about 1.8% efficient. Land area supporting vegetation, it's quite a big land area. The growing length of daylight, 43,200 seconds per day of daylight, averaged, of course. Average 180-day growing season. There's 12 grams of carbon per mole. And if you multiply all these things together, you get about 120 gigatons of carbon globally as what's entering terrestrial ecosystems. So there's no doubt that photosynthesis is extremely important. And photosynthesis is a big reason, I assume, why many of you are here today. Now, as David mentioned, my experiences with photosynthesis started off really leaf scale and and smaller, so all the way down to the chloroplastic scale. And it just so happens that the Farquhar model of photosynthesis, the most mechanistic model that we have to describe photosynthesis currently, is a chloroplastic model. But this model is used for a number of different purposes. Certainly, it's, to fully understand something requires mathematical explanation. So when Farquhar, von Keimer, and Berry got together, Susanna von Keimer was a mathematician. Farquhar was really interested in a lot of the dynamics associated with the influence of stomatal conductance on gas exchange. Joe Berry was interested in trying to elucidate a lot more of the C4 photosynthetic pathway. And together, the three of them, combined each of their individual expertise to pull together the Farquhar model of photosynthesis. And certainly in doing so, they didn't just discover all of these things all at once. What they did was they took a huge pool of information that had been developing over a long period of time and synthesized all that together in a very simple, relatively simple, easy to understand mechanistic model of photosynthesis. And they were so effective in doing that in 1980 that there's well over 1,600 citations, and this model is still being used quite extensively at a range of different scales. Of course, the purpose of modeling photosynthesis for education, it's a big part of what I'm here for today. Fitting data, such as fitting ACI curves in order to get a lot of the parameters that we use in leaf scale or larger scale models. Of course, scaling, when you can model photosynthesis at the leaf scale, it gives you an opportunity then to bring these carbon fluxes from the leaf to the canopy, to the ecosystem, and potentially to the globe. And of course, modeling photosynthesis gives us the opportunity to elucidate opportunities to improve photosynthesis. So by understanding what's driving photosynthesis, we can look for opportunities to improve photosynthesis. And I'll talk a little bit about that throughout this lecture. And if anyone has any questions, feel free to ask. Interrupt me, I don't care. It's fine. So there are many challenges in modeling photosynthesis. The environment surrounding the leaf, the plant, the ecosystem is transient. I mean, right now we've, I don't know if you noticed it, but sunlight just went down a little bit. Some cloud is passing overhead. And photosynthesis responds to that. Temperatures aren't constant throughout the day. When one environmental factor changes, so too many others in certain instances, right? There's uncertainty to what limits photosynthesis under certain conditions. So the fact is that photosynthesis is complex, and the factors that drive photosynthesis aren't uniform. And when I talk about the complexity of photosynthesis, of modeling photosynthesis, it has to do with the fact that there are 3 separate processes that are all involved in giving us a net flux of carbon dioxide. And this is kind of represented here. This is rubisco. This is the primary carboxylating enzyme. This is where carbon dioxide enters the photosynthetic process. So here you have a carbon dioxide molecule from the atmosphere, and this basically, is fixed on RUBP by rubisco, and it forms a carbohydrate. This is grossly simplified. I acknowledge that. Now this carbohydrate, of course, is used in respiration. So you have actually carbon dioxide entering through photosynthesis, and you have carbon dioxide leaving through respiration. In addition to carbon dioxide being taken up by rubisco, so too is oxygen, and that drives photorespiration. When this oxygen is taken up by rubisco, then the end product is carbon dioxide is released. So you have 3 processes. One process takes up carbon dioxide, 2 processes release carbon dioxide, and all this is happening through the stomata. So when you're modeling the gas exchange through a stomatal aperture on a leaf, you're really trying to understand 3 completely separate processes. Admittedly, the CO2 and oxygenase, these 2 activities are related through the rubisco enzyme, which fortunately simplifies the model quite a bit relative to what it could be otherwise. Now, in order to understand how the Farquhar model works, you have to understand a little bit of biochemistry, and I'm assuming that everyone in this room has taken some level of biochemistry at some point in their life. I'm not going to get too involved in that, except to show here that there's this, the Michaelis-Menten response curve is an important component of the photosynthesis model. This is the velocity of a reaction, basically how fast a reaction is going to proceed, and this is the concentration of your substrate. And typical biochemical reactions look like this blue line. And from this blue line, you could determine 2 parameters. One parameter is Vmax, the maximum rate of a reaction, assuming you have an infinite amount of substrate present. So that is the upper limit of how fast that enzyme can catalyze a reaction. And then you have this term, the Km, which is the Michaelis constant. If you take 1/2 of Vmax, and in this cartoon diagram I just made Vmax 1,000, so you take a velocity, a reaction rate of 500, and you just go ahead and extrapolate to what substrate concentration do you need in order to be at 1/2 of your maximum velocity, well, that's going to give you your Michaelis constant. So this is sort of your affinity. The lower your Km, the easier it is for a substrate to find its way into an active site on an enzyme. And the higher your Vmax, generally the more potential you have to carry out a reaction. So Vmax and Km or Ka, some K value, are the really important components of biochemistry that all basically link into this response curve. So I just wanted to give you that overview because it's going to come up a few times throughout this lecture. And by all means, if there are questions, again, interrupt me, because I know that there's probably a wide range of expertise in these types of things. Some of you might actually just be like, well, I'm not really learning anything new today. Others might think that this is so new that I went too fast, and I do have a habit of talking too fast. So if you look at the Farquhar model in its most simple form, that's saying your net carbon assimilation. So this A represents the flux of carbon dioxide between the leaf and the atmosphere. This is going to be a function of Vc, so that's the rate of CO2 uptake by photosynthesis, minus 0.5 VO. Now VO is the rate of release, well actually VO is the rate of oxygenation of rubisco. So you know rubisco can take in the carbon dioxide, it can take in the oxygen. When oxygen is taken up by rubisco, it drives photorespiration. This 0.5 basically means that for every 1 oxygen taken up by rubisco, you get half of a CO2 being released. So in order to get 1 CO2 released through photorespiration, you have to have 2 oxygenation events. And this RD represents the mitochondrial respiration. That's the CO2 flux coming out of the leaf, as a function of that carbohydrate being metabolized by the mitochondria. So basically, if your photorespiration is really high and your mitochondrial respiration is really high, your VC in turn will be low. You end up with negative carbon assimilation. So you're basically just taking the gross uptake and you're subtracting away these two things that are releasing the carbon dioxide. Now, one of the things we want to do when we model photosynthesis is we want to simplify things. And of course, when you're introducing a lot more equations, that isn't always necessarily simplifying things, at least on the surface, but in fact it is. And the first thing we want to do is we don't want to have to separately model three components that are all just additive or, in this case, subtractive from one another. What we want to do is we want to simplify the terms so that we could just basically pull together these first two fluxes. And the reason we can do that is because the same enzyme that's responsible for taking carbon dioxide out of the atmosphere is responsible for oxygenation events. Because oxygen and CO2 are competing for the same active site on Rubisco, we're able to take these first two terms, and basically unify them into one relatively simple model. And the first thing we do to do that is we define this term phi. And all phi is the ratio of oxygenation to carboxylation. Right? So VO is equal to phi VC, just simple algebra here. And you just plug this into the equation. A is equal to VC minus 0.5 phi VC minus RD. So we basically substituted this VO for this value phi. And phi has an intuitive meaning associated with it. And that's presented here. So phi, again, is the ratio of the rate of oxygenation divided by the ratio of oxygenation to carboxylation. And if you look at these terms here, these are those Michaelis constant terms. So VO max is the maximum velocity of a reaction for O is oxygenation and C is carboxylation. And then, of course, we have our affinities for carboxylation, our affinity for oxygenation. And then we have our concentration of oxygen and our concentration of CO2. And this can be simplified even further to 1 over tau times O over C, oxygen over carbon dioxide. The reason why we simplify it like this, because this value here, tau, is intuitively easy to understand. It's specificity. What it represents is how specific is Rubisco for CO2 compared to oxygen. And just to, I think I have a table coming up here, yeah. Just to put this in some context, here's a table showing the specificity of various different Rubiscos from different species and taxonomic groups. And you can see here among the dicots, you can see that they basically average to about 91. That means Rubisco favors CO2 91 times more than it favors oxygen. For monocots, there's a lot of C4s in here, it's lower. So it favors carbon dioxide only 84 times more than it favors oxygen. Now, one thing to keep in mind is that while it would appear that Rubisco favors 90-fold carbon dioxide over oxygen, and that is in fact true, the reality is that the concentrations of oxygen and carbon dioxide around the leaf are not the same. Oxygen is 21% in the atmosphere, CO2 is significantly less than 1%. Right? So it's really not quite as optimal as you might see here. Specificity can actually then be simplified even further into this term we call gamma star. And gamma star is equal to 0.5 times the oxygen concentration divided by specificity. Gamma star is the photosynthetic CO2 compensation point. So gamma star is the point at which the CO2 being taken up by photosynthesis is equal to the rate with which carbon dioxide is being given off by photorespiration. All right? And gamma star is a function of specificity. These two are basically directly related in current atmospheric conditions. Gamma star at 25 degrees is always going to be about 42, and specificity at 25 degrees is always going to be about 90 for C3 planets. If you change the oxygen concentration in the atmosphere, if you're doing some sort of experiment, then gamma star, of course, is going to change, but your specificity will not. So when I say the photosynthetic CO2 compensation point, you really can't find that very easily on an A on a photosynthesis versus CO2 response curve, it's going to be somewhere between the y-intercept and the x-intercept. This basically where gamma star is at the, let's say 42 parts per million is your gamma star. If you just extrapolate this down, bring that over to the y-axis, this first part here represents your mitochondrial respiration, and then this here represents the rate with which CO2 is being given off through photorespiration. So I'm kind of flying through a lot of this. I'm giving you just basic framework, but I'm going to pull it together here right now. So you have this net carbon uptake as a function of your photosynthesis minus what's coming off through photorespiration minus what's coming off through mitochondrial respiration. And then we introduce this phi term, which is a function of the specificity, which can then be used to solve for this gamma star, right? So if we take this A equals VC minus 0.5 and we substitute this VO for this term here, gamma star times VC over 0.5 times C minus RD, and simplify that even further, you can end up with A is equal to VC times this 1 minus gamma star over CI, or C, minus RD. So what this basically means is that in order to model the contribution of CO2 that's coming off of the leaf through photorespiration, all you need to know is gamma star and the CO2 concentration in the leaf. And what you do, what this represents, is it just represents a fraction of the gross photosynthetic carbon uptake that's coming off through photorespiration. And of course, we still have our mitochondrial respiration that's kind of dangling off the back there. So far all I've done is I've taken that VO and I've shown how, in hopefully simplified way, how Farquhar, von Keimer, and Berry back 20, 30 something years ago was able to account for the photorespiration component of this model. Basically by linking the known specificity and the known conditions around that leaf to come up with a way to predict the contribution of photorespiration versus photosynthesis in dealing with the leaf carbon fluxes. So now what we're challenged with is modeling VC, modeling the actual rate with which carbon dioxide is taken out of the air and integrated into the leaf through photosynthesis. And there are a number of different limiting processes that describe this. Environmental conditions are going to dictate what is going to drive VC. The first thing that could determine our rate of carboxylation is how much CO2 or how much rubisco is present in the leaf. And that's what we call carboxylation limited or CO2 limited or rubisco limited photosynthesis. It's been published all three ways. Unfortunately, it's not been standardized. And if you look at the photosynthetic pathway here, this is the Calvin cycle. This is where carbon dioxide enters the Calvin cycle. When you have low photosynthetic rate or low carbon dioxide around that leaf, you're going to be carboxylation limited. If you don't have a whole lot of rubisco in that leaf, you're going to be carboxylation limited. As CO2 concentrations rise, and you have plenty of carbon dioxide around that leaf, it turns out that another limitation starts to present itself. And that second limitation is called light, or RUBP, or electron transport limited photosynthesis. And what that basically reflects is the fact that there's plenty of carbon dioxide, there's plenty of rubisco in the leaf, but there's not enough something after that first event. Something's not in enough supply in order to keep your photosynthetic rates to continue increasing as carbon dioxide concentrations rise. One of the things that could drive that is the amount of ATP or NADPH that are coming from the light reactions. So as a reminder, for those who don't, haven't, I don't know, how familiar is everyone with this? Extremely? What's that? Mixed bag, okay. So the light reactions, the whole point of light reactions is to take solar energy, solar radiation, to drive electrons through the electron transport chain. In the process of doing this, it's performing two tasks. The first task is that these electrons move across this electron transport chain and then pump protons from here, from the stroma into the lumen. And those protons accumulate in the lumen, and there's only one, theoretically, there's only one exit for these protons, and it's through this enzyme known as ATP synthase. As these protons, because there's a proton motive force, there's this gradient in protons from here to here, well, the protons are going to want to make their way out into the stroma. The only path is through this enzyme, and as the protons move through this enzyme, it does a really cool thing. It spins. This enzyme actually spins really fast. And as it's spinning, it's taking ADP and inorganic phosphate from here, and it's converting those to ATP. And that ATP then makes its way into the Calvin cycle and is responsible for replenishing the supply of RUBP. Right? So carbon dioxide and RUBP come together in photosynthesis. That's where it gets fixed. Well, you need to have a constant supply of RUBP in order to accept that carbon dioxide. So this electron transport drives the protons that move this enzyme that makes ATP that then gets shuttled to the Calvin cycle. Eventually, these electrons exhaust the ability to move protons, and they end up being used to reduce this NADP plus into NADPH. And this enzyme, or this molecule here, NADPH, is then also used to provide reductive power to keep the Calvin cycle moving. So the whole function of light reactions are to provide the energy needed to keep the Calvin cycle spinning. And what could happen when you're light or RUBP or electron transport limited is that the supply of energy making its way from the light reactions starts to become exhausted, or it's not fast enough in order to keep the Calvin cycle spinning as fast as potentially it could go. Does that make sense? All right. Don't be afraid to ask questions. I've been teaching this stuff for about 10 years, so I tend to lose sight with, what's intuitive for people to understand. Anyone? No? So I'm an extremely effective teacher, or you already know this? All right. Either way, I'm happy. There's this third limitation. If you'll notice this phosphate, PI here, that's phosphate. ADP, adenosine diphosphate, 2 phosphate groups, has a lot of energy in it. But if you actually add another phosphate group on here, so you have adenosine triphosphate, 3 phosphates, it has significantly more energy. ATP is the most important energy molecule in living organisms. Therefore, this inorganic phosphate is extremely important. Phosphate is not one of those nutrients that are in massive abundance. So recycling of inorganic phosphate is extremely important within living organisms. If photosynthesis is going really fast, right, your rate of photosynthesis is extremely high, you can end up with accumulation of sucrose, and you can end up with an accumulation of triose phosphates. All right, so this is this 3-carbon sugar with a phosphate group attached to it. These triose phosphates are really what go into starch synthesis as well as sucrose synthesis. If it turns out that photosynthesis is going so fast that there are a lot of triose phosphates accumulating, and the ability of the plant to take these triose phosphates and convert them to starch or sucrose isn't able to keep up with the rate with which triose phosphates are forming, then you end up becoming inorganic phosphate limited. So you're limited by how much phosphate is present in the leaf to drive ATP formation. If there's not a whole lot of free phosphate floating around, ATP slows down, formation slows down, your Calvin cycle slows down. We call that TPU-limited photosynthesis. So the challenge of photosynthesis, the first challenge I presented was how in the heck do you deal with oxygenation? And we deal with oxygenation by coming up with, by knowing the specificity, linking that in a gamma star, and then removing some fraction of gross photosynthesis from the equation. When you want to model the rate of photosynthetic carbon uptake, you have to model it separately for each of these 3 processes. So unfortunately, the model involves coming up with an estimate of each of these 3 different processes, and whichever has the lowest rate associated with it is what's limiting photosynthesis under those conditions. So if you look at a photosynthesis or an A versus CI response curve, you end up with something that looks like this. At lower CO2 concentrations, you'll see a rapid increase in photosynthesis. At some point, that increase in photosynthesis begins to taper off, and oftentimes if you take meticulous measurements on an ideal leaf, you can actually measure this inflection quite precisely. At this point, you're actually seeing increases in photosynthesis as CO2 increases, but that's not driven by anything having to do, per se, with more carbon availability. It has more to do with less ability for oxygen to compete with the active site in rubisco, right? So you have CO2 and you have oxygen. They're always competing for the same active site. Initially, as CO2 is going up, more carbon dioxide is available for the for the leaf. You have higher rates of photosynthesis as CO2 increases. At some point, right, you're limited by ATP and NADPH. When that happens, the additional carbon dioxide only acts to provide more competitive power for carbon dioxide over oxygen. And then at some point, your ACI curve becomes completely unresponsive to any increases in photosynthesis, and that's because your triose phosphate utilization limited. In some cases, this can actually go down, and I'll talk about that in a bit. So what we need to do is we have to model basically each of these lines separately. And I'm going to start, and down here, your actual photosynthetic rate is the minimum of carboxylation limited versus electron transport limited versus triose phosphate utilization limited photosynthesis. All right, so back to this curve here. Your rate of a reaction is a function of substrate concentration. You have your Vmax and you have your Km. Now this is for just a simple reaction where you just have a substrate. If you introduce an inhibitor, it's going to influence this curve. But it's only going to influence one of these, Vmax or Km. Who remembers from the biochemistry which one this might be? When you have competitive inhibition for the same active site, will it influence Km or Vmax? Km, that's right. So the way this works is if you add an inhibitor to this and you have an infinite amount of substrate, well, an infinite amount of substrate with a finite amount of inhibitor basically means that the infinite substrate will always outcompete the inhibitor. However, when you don't have an infinite amount of substrate and you have a finite amount of substrate, well, what's going to happen is this Ka, your Michaelis constant, is going to move up because it's showing that the affinity for CO2 is lower, or your Km is higher, they're inverse. Because sometimes when that active site is open, instead of a CO2 making its way in, an oxygen will make its way in. So that's basically going to cause your Km to go higher or your affinity to go lower. So the way you deal with kinetics for a simple enzyme is the rate of the reaction is equal to Vmax times the substrate divided by the substrate concentration plus this Michaelis constant. If you add in the inhibitor, all right, and the Michaelis method derived this decades ago, and I'm not going to drive it. I derived it the first year I was here and realized I was never going to do that again. You need to account for competitive inhibition. The velocity, again, up here, this doesn't change. Down here, the substrate doesn't change, but your Km is altered by this 1 plus the concentration of the inhibitor divided by the Michaelis constant for the inhibitor. So this Km is altered to be higher than, this whole term together is always going to be higher than the Km by itself. Yeah?
Sorry, when you're saying substrate, you mean CO2, and you're saying CO2?
Yeah, so in this case, the substrate, and actually that gets me to right here. So you substitute for photosynthesis. The Vmax becomes Vcmax. The substrate becomes carbon dioxide. Km here becomes Kc. And the 1 plus the inhibitor is oxygen divided by Ko, which is your Michaelis constant for oxygenation. Excellent question. Right? And as it turns out, this is the Rubisco limited photosynthesis model. It's based purely on Michaelis-Menten kinetics. And if you take what we just did and you integrate that into the original equation, remember we got this 1 minus gamma star over Ci from, it's derived in much more detail, but I gave a very general overview early on. So we've already accounted for this. And now our Vc, when you're Rubisco limited, is equal to this Vc max times Ci, or C, divided by C plus Kc times 1 over Ko, or 1 plus O over Ko. Right? So if you want to model Rubisco limited photosynthesis, this is what you need. I should point out that gamma star, you can measure gamma star. There are different methods to getting at gamma star through direct measurements. Most times, gamma star isn't going to change quite substantially among C3 species. It does fluctuate, but not a lot. So usually if you're going to look at gamma star, you could just look it up in published literature. So now we've successfully modeled Rubisco limited photosynthesis. Are there any other questions about that? Fortunately, this is the most mechanistic component of, unfortunately, this is the most mechanistic component of the model. Because from here on out, we start to get just really into more empirical components. And I do want to point out that there's a former student of Steve Long, Jingwang Zhu, who's in China, he's in Beijing, who's actually trying to come up with a unified model of photosynthesis that accounts for every single reaction that occurs in the Calvin cycle as well as on the thylakoid membrane, to try to then understand, instead of just these 2 limiting processes or 3 limiting processes that we've come up with over the last 30 years, is trying to understand, get a better grasp on a mechanistic understanding of what's happening with RUBP limited and TPU limited photosynthesis. As it stands right now, the only one of the 3 processes that we really understand mechanistically well is Rubisco limited. Now, RUBP regeneration limited, also electron transport limited photosynthesis. It's actually in more recent years, it's been going toward RUBP regeneration limited because it's becoming clear that while low light means your electron transport limited, so if you don't have a whole lot of light coming in here to drive this reaction, this electron transport, sunlight's what drives this whole process here. If you don't have a whole lot doing this, then your electron transport limited. But in some cases, it turns out that you might be limited by something in the Calvin cycle that isn't Rubisco. And SBPase is one of them. It's one of the 10 enzymes, 10 other enzymes in the Calvin cycle other than Rubisco. So we're saying RUBP regeneration because it could be under some circumstances that it's actually electron transport that's limiting the amount of NADPH and ATP being formed. In other cases, it could be that there's plenty of NADPH and ATP, but some of these Calvin cycle enzymes just are limiting. Everyone seems to think we have photosynthesis solved. I hear that a lot when I tell people I'm a photosynthesis researcher, and the reality is we don't. We understand the very basic photosynthesis, but what's happening or ways to improve photosynthesis is currently wide open. So, because this is empirical, and given the fact that there's a lot of uncertainty associated with it, we assume that the rate of RUBP regeneration photosynthesis is electron transport limited. That's a general assumption that we make. Rates of electron transport are a function of how much light is available for the leaf, for the chloroplast. The more light you have, the higher your rate of electron transport gets. It will eventually plateau, and when you scale this up to infinite light, you end up with this term Jmax, the maximum rate of electron transport. So, involved in this is use alpha, which is absorptance. Phi PS2 is the quantum efficiency with which light is being used. The efficiency with which light is being used by one of the photosystems. And then beta are the number of photosystems in electron transport. And just to show you what I mean here, linear electron transport, light comes in here. The light splits water into oxygen and protons and electrons. The oxygen and the protons stay in the lumen. The electrons that come off when you break water get excited, and then this high excitation of the electrons is then used to drive there are a number of steps in here. And the energy in those electrons gets lost each step, a little bit of energy is lost from those electrons. But in the process of losing energy, it's pumping protons into here. More light will come in, excite those electrons again in photosystem 1. And when those electrons get excited again, eventually there are a few more steps here, and then those highly excited electrons go into forming an ADPH. The reason we call this photosystem 2 and photosystem 1 is because even though photosystem 2 is the first step in this chain, it was the second one discovered. So there used to just be photosystem, and then someone found a new photosystem and called that the second photosystem. Unfortunately, they didn't find this one first because then it would be a little bit more intuitive. So this requires light and this requires light. So when you go back to this here, beta represents the number of photosystem 1s to photosystem 2s. Half of the photosystems are photosystem 1, the other half are photosystem 2. You assume that they divide the light evenly between them. So that's where we get this beta. Theta is a curvature term. A theta of 1 would show this line comes up, and then basically like a 90, not a 90 degree angle, but a sharp angle. A theta of 0 means that it's a very gradual increase to Jmax. I think that the theta that I set in this model here was 0.8. And that's what we generally assume for theta for plants, but there's some work showing that it changes. Certainly it does change from leaf to leaf, plant to plant, species to species. But in absence of knowing any better, using 0.8 is what the community generally does. Things that can influence theta are the thickness of the leaf, how easy it is for the light to attenuate through that leaf, the arrangement of the chloroplasts, a whole bunch of other factors. Everyone okay so far? So to describe this line here, we use a quadratic equation. This is empirical. Quadratic equation is your rate of electron transport is equal to Q2. It's a quadratic equation. I don't need to read this out. Just for simplicity, so this equation doesn't just go on and on and on, I use this Q2 here. Q2 is the amount of usable light. So it's the total light times the absorptance times the efficiency times the fraction of photosystem 2 to photosystem 1. Empirical equation. Take this. Okay. Take this J, I'm going to do this in reverse order here, times CI divided by 4 times carbon dioxide plus 9.3 gamma star. That gives you, all right, so all this does is it tells you how fast the electrons are moving, your electron transport rate, okay? Knowing the electron transport rate in of itself does not tell you how much carbon dioxide is going into the leaf. All it's telling you is the rate with which electrons are moving across the thylakoid. In order to understand what that means for carbon fluxes, you have to understand what each electron will buy you in terms of ATP or an ADPH, okay? So, the best way to describe this is the electron transport rate will buy you 4 plus 4.66 phi, that phi's come up again, times Vc, okay? Now we've already established that this phi is gamma star over 0.5 times C, right? So we could just substitute that into here. We rearrange this whole equation here to get Vc, your rate of carboxylation, your rate of carbon uptake when your RUBP regeneration limited is equal to your electron transport rate times your CO2 concentration divided by 4 times your CO2 concentration plus 9.3 times gamma star. Now, you guys are all looking at me with blank stares and I don't blame you. This is hard to explain. What this is trying to get at is what will each electron buy you is presented by this down here. Right? So consider electron transport, one electron moving through that chain as a currency. What that currency will buy you depends on the stoichiometry of photosynthesis. And I'll show you what I mean coming up here. All right. I kind of went over this in detail. But basically, the electron transport rate is what takes this NADP plus an ADP, converts it to ATP and NADPH, which then go into the Calvin cycle. And if you look at the stoichiometry here, here's your carboxylation event. This is great. This is what you want. And it produces 2 of these triose phosphates, which then get shuttled through your Calvin cycle. Some of that comes off as carbohydrate. In the Calvin cycle, you need 2 plus 1.5 phi ATP. So if you're looking at the Calvin cycle, this is one area where you need ATP, and that's how much ATP you need. Here, up here at the step in the Calvin cycle, you need another ATP plus phi ATP. All right? And these phi's represent, remember, the ratio of VO to VC. And you need NADPH. All right? This is when you're being this is when things are operating the way you want them to operate. This is what you need. When things operate the way you do not want them to go, so photorespiration occurs or oxygenation occurs, then you need NADPH over here, you need some ATP over here, and you need some ATP over here. So if you take all of these ATP requirements and add them up, you end up with the rate of ATP consumption is basically for each carboxylation, for each VC, each carboxylation, you need 3 plus 3.5 ATPs. For each carboxylation, you need 2 plus 2 phi NADPH. So if you have 0 photorespiration, right, this phi goes to 0. Basically, if you're all carboxylation, all photosynthesis, you need 2 NADPH and 3 ATP. But of course, that doesn't happen in C3 leaves, so these are our requirements. This is, I guess you could say this is the demand. I already went over this. All right. So, rate of NADP consumption, this is our rate of NADPH consumption. That's how much we need based on whatever the environmental conditions are around that leaf, based on a function of VC. If you look at this table I took from Susanna von Kammer's book in 2000, it's basically, there's a bunch of stuff that's going on here, but what I basically want you to focus on is our NADPH consumption, NADPH, right, 1. So for 2 electrons, you get 1 NADPH. As 2 electrons move through the electron transport chain, you will get 1 NADPH. Now we need 2 plus 2 phi NADPHs. Therefore, we need 4 plus 4 phi electrons in order to meet the demand for NADPH. Does that make sense? It's real simple. This is simple. So that's 4 plus 4 phi. When you look at the electron requirements for ATP, it isn't quite that simple. We know that we need 3 plus 3.5 phi ATPs per carboxylation event. All right, that's what we know we need. Now VC, of course, changes just like as it does with anything else based on environmental conditions. Unfortunately, when you're looking at how many electrons you need to produce ATP, there are a number of different scenarios, and we don't really know which is the correct one. So, you know, I mentioned that really super cool enzyme that spins. The reason we know it spins is because someone actually mounted them to a little glass plate, and they attached a molecular rod to the top of it that's fluorescent. And they took a camera and they microscope, fluorescence microscope, and they actually videoed this little rod spinning as they gave it ATP. It was spinning the wrong way, but that's how we know it spins. What we don't know is how many how many protons have to go through that enzyme in order to take an ADP and turn it into an ATP. We know it's somewhere between 3 and 4, but we don't know exactly what it is. It may not be a whole number. It may be. We have ATP synthase in our body, and it turns out that we don't know how many protons in our mitochondria need to go through to produce ATP for us. It's an open question. Not for lack of funding. I think so far, like $30 million has been invested in trying to figure this out, and we just don't know. And we may never know. That makes this a little bit more complex, because when you're trying to figure out how many electrons you need to meet this demand for ATP, you have to come up with a number of different scenarios. So if you assume that there are 3 protons per ATP, right, so that's the lower limit. It can't be less than 3, we know that. If you look at just whole chain electron transport, you look up here, you get 1.5 electrons per ATP, right? And this is how much ATP we need. So you multiply 3 by 1.5 and 3.5 by 1.5. Then our electron transport requirements in order to meet this demand are 4.5 plus 5.25 phi. Whole chain plus Q cycle, I guess that's probably why I put this in. Q cycle is a little cycle that occurs here that actually is more efficient at pumping protons than just linear electron transport. So if you have the Q cycle involved, whole chain plus Q cycle, you get only 1 electron is needed per ATP, in which case you have 3 plus 3.5 phi is your electron requirement for your electron requirement to meet this demand. Now, the problem with these two numbers here, it could be either or, right? Except we know for NADPH, we know that we need 4 plus 4 phi electrons, which means for ATP, this does not give us enough electrons in order to meet the demand for NADPH, right? So not only do we not know how many protons are used to produce this ATP, we have to make sure that whatever we come up with here matches as closely as possible what the electron demand is for NADPH. They're the same electron transport. It's just carrying out 2 functions. So if you assume there are 4 protons per ATP, and you look just at whole electron transport, you can just do the math up here. There are these numbers here in the parentheses. You get 2 electrons per ATP. Then the requirement is 6 plus 7 phi. Now that's just way too high, because NADPH only needs 4 plus 4 phi. This is 6 plus 7 phi. Where's all that extra energy going? It just doesn't work out. So what I generally do is I assume we have whole chain plus Q cycle, where you get 1.33 electrons per ATP. And here's the electron requirement here, 4 plus 4.66 phi. Now that 4.66, that's 0.66 higher than what we need for NADPH. But, you know, it's close enough. It's empirical. So that's how we got the 4 plus, well, that's if you actually, I'll go the other way. That's how we got this 4 plus 4.66 phi. That's the electron requirement necessary in order, that's the, the electron transport rate necessary in order to accommodate the leaf's demand for NADPH and ATP based on whatever your rate of carboxylation is. That was a lot of stuff. You guys all okay with this? Just so you know that I've given this lecture 8 times and I get better every time, so imagine what it was the first time. All right. So if we wanted to pull together the RUBP limited model, again, you have your photosynthetic rates, the minimum of these 3 limiting processes. I've already shown you the your carboxylation limited, which literature assumes is WC. WJ is your electron transport limited photosynthesis, and that's all presented here. So you have your, WJ equals VC when you're electron transport limited. That's just, it's confusing, I know, I apologize. But this is what we came up with, J times C over 4, C plus 9.3 gamma star. J is this quadratic equation here, and Q2, just again, so that the equation doesn't get out of hand, we just simplify that down here. So that's how we model RUBP limited photosynthesis. Questions? That's it? You guys want to take a break? All right. The last thing we need to model is the TPU limited photosynthesis model. This is actually the least understood limitation. And when I say least understood, I mean we know that it has to do with the fact that these triose phosphates accumulate, and the leaf can't use these triose phosphates fast enough to free up that phosphate. So we know what's driving it, but we don't know how it changes with temperature. We don't know what conditions it's limiting. We know that cold temperatures and high CO2 will drive TPU limited photosynthesis, but it's highly variable. So if I measure high, let's say soybean at low temperatures on, one day, I'll see TPU limited photosynthesis. 5 days later, and environmentally nothing seemed to have changed. I won't see TPU limited photosynthesis. So there's just a whole lot of uncertainty with TPU limited photosynthesis. The basic way that people generally use the TPU limited photosynthesis model is basically to assume that your rate of carboxylation is 3 times the rate of triose phosphate utilization, right? So every triose phosphate that's utilized can accommodate 3 CO2s being fixed. And that's because with that one phosphate is attached to 3 carbons. So if that phosphate comes off, then you could get 3 more carbons coming back in, right? That's just, it's not even empirical, it's just complete assumption. There is a more complex TPU limited photosynthesis model that you may or may not see at any point in your long careers. This is a much more complex component, and as I mentioned, sometimes at high CO2, your photosynthetic rate starts to go down. And the reason why it goes down is if you look at, this is the photorespiration cycle, I'm not going to go into this in huge detail. But when this oxygen comes in, I mentioned, there's this 2 carbon chain that's wasted. It's toxic, you got to do something with it. And this is what the leaf does with it. A whole bunch of stuff. There are side chains and everything, or side cycles going on and everything. And then the other product is this phosphoglycerate, which is good. That goes into the Calvin cycle. This phosphoglycolate, the first thing that happens is phosphate is released. And as that phosphate is released, it becomes available, except eventually 2 glycolates leave and 1 glycerate will come back in. When this 1 glycerate comes back in, it actually ties up, this ATP will actually tie up another phosphate. So it balances out, ultimately. But because no cycle is perfectly efficient, as this glycolate leaves, some of that carbon never ever returns. It just ends up being hijacked into another metabolic pathway, which happens. When that occurs, you end up with a net surplus of inorganic phosphate. because this phosphate gets released here, and then this ATP isn't needed to deal with the end product of this whole pathway, which makes that ATP now available for something useful like photosynthesis. As CO2 concentrations go up, photorespiration goes down. As photorespiration goes down, there's less of this surplus of inorganic phosphate that happens. So this model here basically accounts for this A term, alpha, which is the proportion of glycolate not returned. As the photorespiration goes up, this alpha goes down, and you end up seeing this drop you end up seeing a drop in photosynthesis. Now, you don't have to understand this in mechanistic detail, but just know that if you model the simplified TPU model, it's a straight line under all conditions. If you model the more complex one, it looks something like this. And obviously, you can see it's still dropping here. It doesn't drop that much, which is why most people just opt to model TPU that way, if they model it at all. So just to bring this all together, this is the simple equation. This here represents the photorespiration term and the minimum of those three limiting processes. I don't have J written here, it's that quadratic equation, but these are all represented by your Rubisco limited model, electron transport limited model, and your TPU limited model. One thing you may or may not notice is that I divided this by 1 over gamma star over Ci, and that's because when you're TPU limited, you're completely not responsive to CO2 or to oxygen. You're only limited by TPU limitation. So what this does down here is it gets rid of this term right here, and that's actually just as simple as the model is. RD, I didn't even touch on RD. RD is even more empirically based than RUBP or TPU limited photosynthesis. We just assume that it changes with temperature. And people are trying very hard. I know Owen Atkin in the UK, he's still in the, where is he? Australia now, that's right. Owen Atkin in Australia is working really hard and trying to come up with a really good respiration model that can fit into this. Us photosynthesis people tend to recognize that RD for most systems is so low that you can have a significant amount of error in RD when you're modeling leaf level photosynthesis that it's not necessarily worth the effort to try to come up with as mechanistic model as possible. But more recent research is challenging that. And that's why there are now people who are pursuing this, right? So there's still a lot to learn in that. These parameters are what you need to model photosynthesis. There are certain parameters we assume to be constant or similar for all C3 plants, even though we know they are not. It turns out that for most C3 plants, they don't deviate enough from one another for us to make a big stink out of it if you're trying to model leaf, plant, or canopy photosynthesis. If you're trying to understand the influence of different growth environments on Rubisco, then you need to measure these types of things. Right? So it all depends on what you're doing. But if I wanted to go out there right now and model an aspen canopy, an aspen ecosystem, I'm just going to go ahead and see, you know, what's been published in KC, KO, and gamma star previously, and I'm going to assume that those kinetic parameters are going to apply to my, you know, aspen system. Variables, parameters that are variable across all conditions and you really need to know are VC max. That's a function of how much Rubisco is in the leaf and the activation state of the Rubisco in the leaf. Jmax is a function of how much electron transport capabilities that leaf may have. RD changes based on a leaf. I know a lot of people assume that's always a function of VC max, but it necessarily isn't always that case. If you're going to model TPU, you need to know what that is. I threw GM in here. I don't know why I did that. It's mesophyll conductance, and I'll get into that in just a little bit. Some of the work that I had done early on for my PhD was to come up with the most accurate as possible estimates of how each of these parameters change with temperature. So this here is the temperature response of gamma star. All right, and I think this dotted line represents what was assumed previous to my analysis, and this is what I came up with afterward. This temperature function of gamma star for modeling leaf photosynthesis for C3 plants can just be plugged into whatever model you're using. If you want to know specifically what's happening with your leaf, you think your gamma star differs from what I measured on these tobacco plants 12 years ago, then you can measure gamma star for your leaf. I mean, it's entirely up to you. Most people who model tend to assume that the variability driven by the actual gamma star versus the gamma star derived in this paper is so small relative to the variance associated with uncertainty associated with modeling that it just makes sense just to use those. Temperature responsive RD was here. VC max changes with the temperature in a very predictable way, but VC max differs from plant to plant, leaf to leaf, so on and so forth, time to time within a leaf. Therefore, I came up with a temperature correction of VC max that's normalized to 1 at 25 degrees centigrade. So if you go out and you measure an ACI curve on a poplar tree, and you find out your VC max is 30 at 25 degrees, you can apply this function and see that it's, whatever, 120 at 35 degrees, or, 14 at 15 degrees, or whatever it is, based on this temperature function. Kc and Ko temperature functions are here. These, again, are assumed to be constant among C3 plants, so you could just integrate these into your model. I do want to point out that these temperature functions have been applied to, they were derived from antisense rubisco tobacco plants. And they've been applied to poplars, they've been applied to desert shrubs, they've been applied to individual chloroplasts, they've been applied to a whole range of different ecosystems. And they turn out to do a pretty good job. If you're trying to show differences in the kinetics of plants, of rubisco kinetics of a plant from, let's say, a xeric to a wet habitat, you want to go ahead and you want to find out these parameters independently. Right? So it all depends on what you want to do. If you're doing ecosystem modeling, there's no point in reinventing the wheel. If you're doing modeling of a very specific plant that you think deviates in its rubisco kinetics from most C3 plants, then you want to figure those out on your own. What we're going to do is we're going to model photosynthesis using an Excel spreadsheet. Here are just a couple of things that I've done with the model, showing this is actually a recreation of a seminal paper that came out in 1991 by Steve Long, where he showed the importance of CO2 and temperature together. And he used the Farquhar model. He used it with its original parameterization, which didn't actually have any temperature corrections built in, but rather they were just assumed. And it showed that as temperatures increase, the CO2 effect becomes even greater relative to the lower CO2. So you see the CO2 by temperature interaction, highly predicted by the model. If you look at, well, I don't know why I put this in here, RD. I just took this from a paper, that's why. And if you look at the function of temperature, the model, using the model to come up with how various different processes or different inputs of the model change. You can see, here's a graph that was in a chapter I published with Tom Sharkey. So this is specificity as a function of temperature. Specificity is this line here, and you can see that at really low temperature, your specificity is extremely high. Low temperatures favor carboxylation over oxygenation. High temperatures favor oxygenation relative to carboxylation. So your specificity goes down. As your specificity goes down, this phi value goes up. That means the ratio of oxygenation to carboxylation goes up. And this 1 minus gamma star over Ci, that's the fraction of CO2 being fixed by photosynthesis being released back out, goes down. Right? That means that you're getting more CO2 being released through photosynthesis. The reason why I'm showing these is because what I'd like you guys to do is, using this Excel model, is to try to recreate a lot of these types of things. In order to do that, what the Farquhar model does is it provides the demand function. Right? So that's the demand for carbon dioxide. That's basically your photosynthesis model. It's these black lines here. Right? The supply function is how much CO2 is present, and the supply function is going to be related to the barriers of diffusion for CO2 to make its way from the atmosphere into the leaf or into the chloroplast. All right, so Ci is your intercellular CO2 concentration. That's your CO2 concentration in the air spaces within the leaf. Right? And that's determined by your boundary layer conductance across the leaf, and it's determined by stomatal conductance. If you want to determine, actually, if you want to model photosynthesis based on what's in the chloroplast, then you need to account for your mesophyll conductance. And that's the diffusive barrier from those intercellular air spaces to the site where carbon dioxide is taken up by rubisco itself. So if you model an ACI curve just based on CI, you end up with this first curve here. If you model it based on CC, then your curve shifts this way, right? Most people generally aren't modeling based on CC when you're looking at anything related to the leaf, plant, or ecosystem scale. But there are instances where modeling CC is important. And I don't, for the sake of time, necessarily want to get into it. But this here just reflects the resistive pathways going from the atmosphere. You have your boundary layer resistance or conductance, which is a function of wind, the size of the leaf, shape of the leaf, the surface of the leaf. All those things are going to influence your boundary layer conductance. Your stomatal conductance is going to be a function of how much stomata, how many stomata are present, as well as the size of opening for the stomatal aperture. So leaves have a lot of control, plants have control over how open or closed the stomata are. So there's a lot of biological control involved in stomatal conductance. Mesophyll conductance is very likely just a physical phenomena that's driven by changes in the thylakoid membrane or in the cell membranes, chloroplast membrane, as temperatures change. There's also some theory that aquaporins or carbonic anhydrase might be involved in that. But the fact is, mesophyll conductance, we know it changes with temperature, we know it changes to some extent with CO2. What we don't know is mechanistically what's driving changes in mesophyll conductance. Modeling CA does not account for the impact of stomatal conductance. So if you're modeling an ecosystem, you do not want to model that ecosystem just based on CA, on your atmospheric CO2 concentration. Because stomata are dynamic. They change with light, they change with temperature, they change with vapor pressure deficit, they change with a whole range of different things. So if you account for CI, you're not accounting for the impact of mesophyll conductance. Mesophyll conductance also changes, but the original model that was presented with the temperature functions that I gave in 2001, already it's an in vivo model. So those changes in mesophyll conductance, assuming they're similar from species to species, is built into the model. If you want to actually model as precise as possible what's happening in the chloroplast, then you can model on CC, but then you actually have to have a reliable way of measuring mesophyll conductance, and that's not a trivial thing to do. In order to model the Excel model that we're using, or if you want to model photosynthesis, you have to model the supply of CO2 going in. The ball-bearing model is an empirically based model. It was actually changed or updated by Looning. I did not have this in my first year here, and Looning was sitting right here, and he let me have it. Unfortunately, he's not here this week, but I still, if I had known ahead of time, I would have taken this out. No, I'm just kidding. But basically, you can model stomatal conductance, and you have a GO, which is your stomatal conductance at light compensation point. You have these scaling coefficients, alpha, and then D0, D naught. And then you have your CO2 compensation point, not gamma star, gamma. That's actually where your CO2 going in the leaf matches going out of the leaf. Gamma reflects photosynthesis, photorespiration, and mitochondrial respiration. Gamma star is the CO2 photosynthetic compensation point, which is just photosynthesis and photorespiration. So you basically look at this, but basically stomatal conductance goes up with photosynthesis, it goes down with increasing vapor pressure deficit, and it goes down with higher CO2. So this is the model that's basically used for modeling stomatal conductance. Unfortunately, A and CO2 at the leaf surface are needed to model stomatal conductance, yet A is what's used, is what we're shooting for in modeling photosynthesis. So the only way to actually model photosynthesis in a defensible way is to do it iteratively. Because here you have A that depends on CI. Well, CI is a function of, this is just your fix analogy, is a function of A and GS is a function of A, which you then need A to get CI, or you need CI to get A, you know, I could just keep going. It's circular. So the fact is you have to solve for this iteratively, right? There's no other way to do it. And with an Excel model, we can't do that. So as you're using the model later, you're going to find that there's a very basic, simple stomatal conductance term or equation built in there that's even worse than the highly empirical ball-bearing model. I also want to point out that there are a lot of people working on a mechanistic model for stomatal conductance, and the math that gets involved in there is tremendously, well, it's inhibitive to many people, myself at this point included. And these models have yet to really be fully validated, but you'll probably learn about them in the next 5 or 10 years. You know, I think there will be a big breakthrough coming up. Now I want to talk about what we're going to be doing at the computer lab. We're going to be doing 2 separate activities. The first activity is using a very simple curve fitting model that I came up with back in the early days of my grad school career, where I needed to get VC max and J max from ACI curves. Now yesterday you took ACI curves with Pat, and today what I'd like you to do is I'd like you to try to fit those ACI curves into this spreadsheet to try to get VC max and J max out of it. There are much more statistically rigorous, defensible, objective ways of getting VC max and J max that you want to use if you're going to try to publish anything on VC max and J max. You do not want to use this spreadsheet. This spreadsheet is an excellent learning tool. It is not an excellent research tool. It had its day, but now with SAS or with R or with MATLAB, there are much better ways of fitting things that remove all subjectivity from the curve fitting. All right. I do want to point out that I reanalyzed all my data using the SAS curve fitting thing, and it actually came up with the same answer that I came up with on my own. So that doesn't question any of my previous research. But there was subjectivity involved in there, and I consider myself to have been pretty lucky. What you want to do is there are going to be 3 sheets in this PS Fit program. The first sheet here is what you come up with. This is where you input data. So temperature, all points that I made for this might have been a fake ACI curve, were done at 25 degrees centigrade. The next column is your photosynthetic rate. The third column is your CI. All of these come directly from the Lycor. And the fourth column is the CA. That's the CO2 in the sample chamber. And when you put these in, you also want to enter the CO2 concentration. That's in millibars, so it's going to stay at 210. Assuming, well, yeah, it's still 210 up here. I mean, it's lower pressure, but that doesn't matter because it's millibars. Light level you used, and leaf absorptance. Leaf absorptance for these trees outside right now is probably 0.85. That means that they're intercepting like 85% of the light, absorbing 85% of the light. But the Lycor system uses a red-blue light source. Is that what you used? Yeah, red-blue. The leaves will actually will absorb red and blue more efficiently than they will red, blue, green, the whole spectrum, right? So that actually absorptance is 0.93 if you're using the Lycor red-blue light source. So you put that in. This will show you whether or not your curve looks good. It should look something like this. Some of these trees, as I mentioned, have low rubisco, so you may never actually even see RUBP limited photosynthesis pop up. I don't know. Depends on what they look like. So then the second thing what you want to do is you want to add all of the data from the previous sheet. You want to copy and paste it, not cut and paste, but copy and paste it here. And what you want to do is you want to keep deleting values from the higher CO2 concentrations until either your R-squared approach is 1, or until you'll see as you have them all in here that you'll see some come up in a straight line, and then they'll branch off like a little flag up here. You basically want to keep deleting them until when you delete the 1 point, if we were to delete this 1 point here, your VC max, this line would, instead of shifting that way, might shift that way, or it won't change. Right? So this is the subjective part of this thing, is you keep deleting the points that don't fall in a straight line until all you're left with are the points at the lower CO2 concentrations that end up in a very linear relationship. And all the points that you then deleted from here, you copy from the first page and paste in here, and you should end up with something that looks pretty straight here, right? And basically, like I said, this was done for my own personal analysis 14 years ago. But it actually turned out that there was a large community of people who use this, and now it turns out that it's a good learning tool. So you should end up with a line that's straight here and a relatively straight line here. You can see we might be getting into some TPU limitation here because it's kind of branching off, but I'm not going to worry about that today. The third sheet then will take all this information and it'll draw a red line, or I guess a maroonish line, to the Rubisco limited and a red line to the RUBP limited. And what you should see is that the solid lines overlay the actual measured data as close as possible. And then if you look up here, you'll see your VC max. Your VC max corrected to 25 degrees, assuming you didn't collect them at 25 degrees. Your J max and your J max corrected to 25 degrees. Again, assuming you weren't measured at 25 degrees. So what you're going to do to start with is you're just going to get some values for VC max and J max from the spreadsheet, from the data that Pat and you collected yesterday. And then once we spend about maybe 45 minutes on this, we'll turn and start actually taking those values, putting them into the Farquhar model in another spreadsheet, and then you can just start running a few scenarios. If the website's up and running, this is what today's website looks like. Down at the bottom, you'll see that there's PS Fit 7.3 link, and that's actually what you're going to be using initially. And then the Farquhar model F, that's not written in some crazy F programming language. That just means that that was the final version before I left it. So these are what you're basically going to be working with today. Any questions?